I will start sharing the screen. Can you see the
Yeah, almost. Yeah, now
screen? And we basically use the slideshow.
Yeah, it's good.
Right, and maybe I even try and get that laser pointer up. Okay, so if we're good to go, thank you very much, Manu, for the introduction. Manu and I are actually collaborators in Leuven, so it's a real pleasure for me to be introduced by Manu. Thank you for the invite to come and talk about our paper today. I feel a little bit of a cheat being a PI and presenting, but when I got the invitation, or my former student got the invitation to present and forwarded it to me, I was struck by the quality of the other presenters and really just wanted to join in. Okay, so as Manu said, my lab is very interested in molecular mechanisms of astrocyte function. And when I originally joined the lab, it was actually to look at mechanisms underlying gliotransmission. And as I'm sure many of you realize, this is actually quite a polarized debate now. It's unclear whether it occurs via vesicular mechanisms, for instance, or non-vesicular mechanisms. And one thing that struck me very, very early on was the potential for heterogeneity to play a key role in this. And then I think, as Anna so nicely illustrated in her talk, when you actually consider the whole range of functions that astrocytes are now thought to perform, it really becomes a question of whether one astrocyte can perform all these functions or whether specialized subtypes exist. So when I started my lab, it was already known that there are regional specializations of astrocytes which are important for cell survival and function. But what was missing at the time, and what we thought would be useful, was a complete molecular census of astrocytes in defined brain regions. And that is really why we started the project, to get a handle on basically what makes a cortical astrocyte, for instance. So today I want to talk to you about the paper that recently came out. I want to introduce you first of all to the methodology that we use, give you a brief overview of the results, our single cell analysis, the in situ hybridization validation, and also some functional work. I will skip basically through conclusions, and then I would like to spend a few minutes introducing a web resource we developed, which I hope that people in the astrocyte community will find useful. If any of you are interested in learning more, the paper, as advertised on the flyer, was recently published in Nature Communications. I would also point out a further follow-up study that we published together with the lab of David Rowich, which was also published this year in Nature Neuroscience. Okay. So when we talk about heterogeneity now, I think most people's idea automatically moves to single cell sequencing, which is probably the easiest and most comprehensive way to get an unbiased view of heterogeneity within a cell population. This is the approach we took. All the work that I'm going to show you today was actually done in P56 black 6 mice. Why? Well, black 6 is a common lab mouse strain. P56 not only avoids developmental issues that are known to occur in astrocytes, but it's also age matched to external resources such as the Allen Institute database. We decided to concentrate on two areas, cortex and hippocampus. This is partly because we do a lot of in vivo imaging through cranial windows, which of course is superficial cortical layers, and because we have an interest in the department in Alzheimer's, in which hippocampus is a particularly vulnerable region. So the way we do the experiment is we basically take these two regions, we do a mechanical dissociation based on enzymatic digestion. We remove excess myelin using Percol, and then we selectively mark astrocytes using the AXA2 antibody, which we know binds to the ATP1B2 protein on the surface of astrocytes. And we also exclude heavy myelin debris by simultaneous staining for O1. We then FACS sort the cells, and we deposit one cell per well in a PCR plate, which is actually harder than you think to do initially. The protocol that we chose was based on SmartSec2 technology. This will be quite different to the so-called 10x technologies most people are using nowadays. We chose SmartSec2 because it is full-length transcriptome coverage, meaning that we can also look for differential promoter usage and alternative splicing. And it's also extremely sensitive, okay? Much more sensitive than we found the 10x to be, at least in the early versions. Having said that, astrocytes contain round about 1/10 of the RNA of a typical neuron. So we had to optimize the protocols for use specifically in astrocytes. For those of you who are technically minded, this meant that we had to change the concentration of template switching, oligonucleotide, and PCR preamplification cycles. Okay, we sequenced close to 3,000 cells. Oops. Okay, and of those 3,000 cells, which were sequenced with a read depth of approximately 1 million reads per cell to get optimal coverage, 2,031 libraries passed quality control. So these are metrics such as GC content of transcripts, 5', 3' bias in the reads that are detected. Okay. Now, I would like to point out that most single cell studies actually do employ these metrics, and you need to keep them in mind because they are purely computational reads, and we actually define the cutoff points for this work. Okay. And what we found when we looked at the libraries was we had close to 2,200 genes detected per cell. So when we take that information and we put it into SURAT, which is one of the industry standard analysis pipelines, what we actually find is the following. Okay. So SURAT detects higher order cell types, as can be seen here. For those of you who are not familiar with this type of data presentation, this is a so-called t-SNE plot. It's basically just a way of reducing very high dimensionality data to a two-dimensional representation. And essentially the take home is cells that are alike are positioned close together. Cells which are, not very similar appear quite far away. So as you can see, the majority of the cells we actually sequenced were astrocytes. We identified a small amount of contamination based on known marker genes, and these cells were discarded from further analysis. So we took 1,811 astrocytes, which expressed 886 variable genes, and we reclustered again using SURAT. And what we found was 5 major astrocyte subtypes, which can also be seen here in just a very simple blot of cells versus transcript number. One thing that I would like to point out is that for those of you who are familiar with neuronal single cell sequencing, you get very, very obvious differentiation very, very early on in neuronal signatures based on characteristics such as neurotransmitter phenotype. This doesn't occur with astrocytes. Astrocytes seem to be much more complex from a single cell point of view with multiple axes of heterogeneity, okay? Now, despite one of our concerns actually when we saw this was the possibility that, we were introducing artifacts during preparation of independent cell batches. So we performed a lot of analysis for this. Suffice to say that none of the experiments we performed showed any bias. The one bias that we did see was already reported, which was a clear separation of cortical and hippocampal astrocytes. Okay, so this gave us a lot of confidence that our single cell analysis was heading in the right way. Okay, now when we look within those astrocyte subtypes, it was possible to distinguish a number of molecular markers which basically act as molecular signatures for those subtypes, which I will come back to in later slides. So when we look at the data, as I said, astrocytes express close to 2,200 genes, maybe 2,500. Oops. Okay. What is very interesting, though, is there do seem to be a set of common genes expressed across astrocytes. So in this case, we routinely find about 350 genes in at least 60% of astrocytes. And these encode for core functions, which we would associate with most cells, specifically astrocytes such as glycolysis and gluconeogenesis, OXPHOS, cholesterol synthesis for synapse formation, lactate metabolism for power generation, and also more recently, circadian function as shown by Mick Hastings in Cambridge. What we got quite excited about, though, from a cursory inspection of the data, was despite the similarities, there are also major differences. And these major differences are most apparent at the level of transcription factor expression, okay, as you can see here between the different subtypes, implying that there are different gene regulatory networks at play in each of the subtypes, which influence presumably the development, but ultimately the function of astrocytes. Okay. Now, of course, everything that I've shown you on the slides up to now is purely an in silico analysis. And the big question is, how does that compare to the in vivo situation? Okay. Now, luckily, when we actually took the hippocampus out for our tissue preparations, we also took the dentate gyrus. And what we noticed in subtype 4 is that they are highly enriched in transcripts which encode or are thought to be associated with processes such as mitosis and cell cycle regulation, transcriptional activation, neurogenesis, and neuron differentiation. Okay, so the question is, are these actually astrocytes that are found in the dentate gyrus? So to investigate this, we basically turned to multiplexed in situ hybridization. Here you can see the results of a staining we did for subtype 4, which is marked by the subtype marker frizzled B. And we use SLC1A3, which encodes the glutamate transporter GLAST as a general astrocyte marker. Okay. So you are going to see this type of presentation quite often in the next few slides, so I will just run through it in detail. What you're actually seeing is the coronal section of P56 mouse brain. The subtypes of interest are marked in black dots in the low magnification images. Below, you can see higher magnification images which show co-localization of these markers to individual DAPI-stained nuclei. On the right-hand side of the coronal section, you can actually see how the cortex and hippocampus are subdivided based on classifications from the Allen Brain Atlas. And when we decided to quantify the data, you can see the results on the right-hand side of the slide. In this left-hand column, if you like, you will see the raw transcript counts for each cell identified in an area of interest. In the middle column, you actually see how the astrocytes for a given subtype distribute between the various layers of the cortex and hippocampus. And in the right-hand side, or column, you actually see the proportion of the subtype in relation to the total astrocyte content of a particular region. And as I hope you can appreciate, most of subtype 4 cells were actually found in the subgranular zone of the hippocampus, which is consistent with a role as progenitors, giving us lots of confidence in our single cell data and also our ISH-based techniques. Okay, so building on that, we went through all the subtypes to get an idea of where they were situated within the tissue. Subtype 1, which is basically defined by the expression of GFAP and AGT, there were no surprises. It was expressed mostly in the peel layers and hippocampus, which is consistent with immunohistochemistry data. And there were no real surprises when we actually looked into the gene ontology analysis for the transcripts that are expressed by this cell type. The only real areas of interest were differences in transcripts associated with synaptogenesis and neurite outgrowth, as well as it appears that astrocytes in these regions are specialized to support glutamatergic neurotransmission. Subtype 5 was actually the rarest subtype that we found. It is basically marked by an absence of OGT and the presence of FAM107A. It's interesting because it shares a lot of genes in common with subtype 4. Approximately 14% of genes to 25%, depending on how you do the maths. It's also randomly expressed through what seems to be the cortex in the hippocampus. Okay, and as well as showing genes that are involved in cell proliferation and neurogenesis, it actually starts to show genes that we associate with mature astrocyte functions, such as OXPHOS and glycolysis, and also cell-to-cell interactions, okay? Which makes us hypothesize that actually this rare subtype is actually a rare progenitor that you find within the tissue. And one area of interest that we currently have in the lab is, are these cells actually, the precursors of subtypes which later undergo rapid proliferation in the cortex, for instance, to produce the mature astrocyte population. Subtypes 2 and 3 are interesting because they are distributed throughout the cortex. AST2 is mainly seen in cortical layers 2 to 6. Subtype 3 is also seen in layers 2 to 6, but you can also see expression in layers 1 and also really quite high levels of expression in hippocampus. And the reason we find this interesting actually is because when you look at the gene ontology for transcripts detected, they actually show extreme differences. It appears, despite the fact that they are in the same cortical layers and are, to the best of our knowledge, intermixed, differences in specialization to support either glutamatergic neurotransmission or GABAergic neurotransmission, and also even initial synaptogenesis and neurite outgrowth, okay? Implying that local environment could play a significant role in actually tuning the molecular identity of mature astrocytes. Okay. So we have obviously molecularly distinct astrocytes. We know from previous work that's been published that astrocytes show morphological differences, which I don't want to go into. The question is, do our subtypes actually show functional differences, which may impact on local neurons? Now, this was actually quite a difficult question that the reviewers raised when we submitted the paper initially. Most of the markers we use, there are no good marker lines available. most of the markers encode intracellular proteins, which make it very difficult to fact sort those cells for cell culture-based experiments. So we actually took a different approach in which we basically thought if we sample enough cells in the different brain regions, we will get a handle on whether there are differences in function. Okay. So the following slide actually shows how we did the experiment. Basically, we load astrocytes with fluo 4AM, which is a calcium indicator, and we identify astrocytes based on the cell type specific uptake of sulforhodamine 101. We then locate the cells in specific areas, whether that's CA1 of hippocampus, layer 1 of cortex, or layer 3-5. And we record calcium transients in those cells, both under conditions of baseline activity, when they have been isolated from local neuronal activity by application of tetrodotoxin. And finally, after application of the alpha-1 adrenergic agonist phenylephrine, which basically invokes calcium transients in the majority of astrocytes. And as you can see, when we look at cells and their calcium activity based on basically calcium increases in fluo4 signal above baseline, you can see that cells in the different regions express or show calcium transients that have remarkably different properties. I should also like to point out actually that all the cells that you're going to see quantified in the next slide were basically retained for analysis only if they showed a response to phenylephrine. Okay. Now you can obviously characterize those calcium response curves in a variety of ways. You can look at the mean amplitude of the response. You can look at the mean peak width. You can look at the frequency of responses, the mean peak time, for instance, the area under the curve. And the take-home message is that there are significant differences in calcium signaling in astrocytes across brain regions. Okay, and these differences map more or less actually to where we find these specific astrocyte subtypes, right? Implying that these subtypes not only show distinct molecular profiles, but also functional activity. Okay, so to conclude, I hope that I have shown you that not only do astrocytes show interregional heterogeneity as previously thought, but they also show quite substantial intraregional heterogeneity. Okay, and I also hope that I've shown you that it is actually worth optimizing a protocol when you want to specifically investigate a particular cell type, because the so typical generic type protocols that have been used in studies to date, we basically think just weren't optimal for astrocytes. Okay, those subtypes are defined by unique molecular signatures, spatial positions, and calcium signals, consistent with really great work from colleagues. What is actually interesting is that this implies that there is micropatterning in the cortex. And actually, in a follow-up paper that we did with the lab of David Rowich, we actually go on to show that not only do we see, you know, astrocyte subtypes within cortex, but when we analyze further, we can actually see further breakdown in those subtypes, and that there actually is specific cortical lamination of astrocytes, which we think basically is responsible for differential synapse formation in the different layers of the cortex. Okay, so my lab is still quite young. We have basically taken this data and we are running a number of projects now within the lab based on it. One project that I am super keen on is the potential for differential excitatory versus inhibitory synapse formation based on the local astrocytes. So if any of you are listening and are excited about this, then we actually do have space in the lab and funding to pursue this. And I would be super happy if someone shared my excitement for this. We do hope that it will also benefit the entire field in that it will help, you know, identify new markers for astrocytes, even subtypes, help the development of new Cree lines, for instance. One final word, if I have time. We regard this as a resource for everyone. We're not going to keep the data to ourselves. As part of the publication, we actually developed a database which is hosted by my lab. All the raw data, gene counts, ISH data, everything, not only from this project, but follow-up projects we've done on the effects of disease on astrocyte subtypes are all contained here. So if you're interested, please go and, you know, basically help our numbers for our future grant applications when we cite that this is useful. And last but not least, thank you to the people who did the work. Particular thanks to Arax Martirosyan, who did all the single cell bioinformatics. She was helped when she first came into the lab a lot by Grant Bellegarde and Chris Ponting in Oxford. Thank you very much to Jerome Verhees, who did all the functional analysis, basically as a side project in the lab for him. And also, like I said, I would just like to thank Omer and David for the follow-up paper we had, and also the people who pay for my lab, particularly VIB, my host institute, KU Leuven, and the European Research Council, who actually gave me the money to start astrocyte research. Last but not least, thank you all of you for listening, and I'd be happy to take any questions.
Thank you, Matthew, for the very interesting talk. So we have some questions. Is it that possible that the AST5 astrocytes will become neural progenitor cells under some pathological conditions?
It could be. It could be. Actually, one of our follow-up projects, which we're just about to submit, is actually looking at traumatic brain injury. So, you know Pascal, of course. So we did a single cell sequencing study in collaboration with the Fitzsimmons lab in Amsterdam. And what really looks to change in the brain after a traumatic brain injury actually is the gliogenic and neurogenic potential. What we actually see is a massive drop in gliogenic potential in the brain, and we see basically a switch to the brain almost trying to compensate for neuronal loss by increased amplification of neuronal progenitors, as if it's trying to push the generation of more neurons to replace damaged circuitry.
We have another question. Do you think the astrocyte heterogeneity is developmental or acquired environmentally?
Actually, I sit on the fence on this. I think both. I think that if you look in spinal cord, David Rowich has, you know, over the years done beautiful work showing the effects of, you know, predetermined developmental programs. Keith Murray recently with Pharma had a beautiful paper showing that neuronal release of Sonic Hedgehog was responsible for, you know, basically setting a transcriptional state in cerebellum, I think it was. There have been other studies looking at knockout of neuronal V-glutes, which influence astrocyte size and so on and so forth in the cortex. Denise Garcia's shown, you know, heterogeneity in sonic hedgehog receptors. So as I said, I sit on the fence. My favorite hypothesis is that, you know, astrocyte subtypes 2 and 3, from a developmental point of view, are identical. But this differentiation to support glutamatergic or GABAergic transmission actually is at least partially pushed by local environment in the cortex.
So we have one more question. How stable are these signatures for an individual astrocyte? Is it on the same line?
Yeah, sure. It's a very good question. We get it all the time. What's the difference between a subtype and a cell state? One thing I think everyone always has to consider with single cell data is they are a snapshot of a mouse at a given age on the particular day you killed it. Okay. Having said that, we have, you know, looked for these subtypes over tens, if not hundreds of animals using in situ hybridization. And we see that they are present and robustly from animal to animal, irrespective of effects such as gender. One thing that we've not looked at, which I think may be interesting actually, is the effects of circadian time, because all these animals were killed, you know, at the same time in the morning. But essentially the only manipulation that we have made in the lab which shows any form of effect on these subtypes is actually inducing acute injury. Okay, and the injury that we are most fond of is traumatic brain injury. And with that, we do see changes, but in a normal, healthy animal, they seem to be very, very robust.
Okay, thank you, Matthew. So thank you for your nice talk, and I will leave the word to Jerry